Okay, guys, I'll go over these briefly as well. Other types of biotech. So some of this we've already spoken about already. And in the next few slides, in the master ag notes that I found, they give some more detail and different examples. So we'll just go through that. So another biotech would be biofuels, which we've discussed. An example would be algae. And how's that used in Australian agricultural production? Well, algae can be successfully used to manufacture all biofuel types. Algae does not compete for land, for food production, and some types can be grown in saline water, so salt water, and so has the potential to not compete for agricultural water, so you don't need to use fresh water. Algae show great potential as biomass for biofuel for agricultural machinery usage. Algae byproduct can also be used as a protein source in stock feed or as a fertiliser, so a whole heap of benefits for that. Biopesticides, again, we've spoken about this at length, BT cotton. Plant incorporated protectants, PIPs, pesticidal substances that plants produce from genetic material that have been incorporated into the plant. Examples include Bacillus thuringius bacterium, which has been incorporated into BT cotton to disrupt the cotton bollworm Helicoverpa or Megera. PIPs allow for biological treatment of specific pathogens. The use of PIPs has many advantages for production in terms of financial savings in labour, chemicals and production losses, as well as reducing negative environmental impacts associated with chemical pesticide use such as runoff and soil/water contamination. So hopefully you're seeing a bit of a theme here. BT cotton is coming up a lot in terms of how you can use it for different answers. So I've definitely learn everything you can about BT cotton. I know it might seem a bit extreme with the Latin terms in there, but that's an extra few marks if you can remember them. But don't spend your a half a day trying to remember the Latin names. But if you can remember BT cotton, even Helicoverpa as the cotton bollworm, if I was a marker, I'd be throwing your marks. Rumen modification. So we know ruminant animals produce a lot of methane, so cows. The MLA's National Livestock Methane Programme using algae-based functional foods for reducing enteric methane emissions from cattle, so stop them burping basically. The use of specific varieties of macroalgae at precise quantities and combinations has proved to have commercial potential to increase feed conversion and efficiency, thus potentially increasing cattle productivity whilst reducing methane emission. So we know that the production of methane as a greenhouse gas is a real problem with cattle. So we can reduce that by feeding them something different. Then that's awesome. That could have a really positive environmental impact. Gene markers, SMP chips. For example, the ovine SMP50 bead chip. So the gene marker is just areas where we can say, okay, that's where this gene is on the DNA. So DNA is very, 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 very long. So if you can have markers that point you in the right direction, then that's going to help save you time and you can manipulate that gene when you want to. So why is this beneficial? Well, it can assist in selective breeding and breed performance recording, reduces time and genetic gain through identification of organisms with desired genes to be used for future breeding. So if you know where those genes are, you have a gene marker on that part, you can then just replicate that gene or turn it on, turn it off, whatever you want to do with it. Vaccine production may not seem like an obvious biotech, but obviously these are improving all the time. An example would be beef research into cattle vaccinations using novel anti-cattle tick antigens to get rid of ticks. So research has found solutions to develop commercially effective cattle tick vaccinations. Cattle ticks have direct negative effects on productions, slow growth rates, anemia and death, risk of market loss through chemical residue contamination, and the cost of tick control. Development of a commercially effective vaccine would increase on-farm and industry productivity and profitability. So we know about drenching and we know about treating the cattle for various ailments, but if you had a vaccine, then it's going to prevent some of these problems and reduce the amount of chemicals we need to use in the cattle and also reduce the costs. Embryo testing or sexing we spoke about for the ethics involved in that. Embryo testing for the MOET programs to grade embryos and remove degenerate embryos or unfertilized ova. So that's just an example, different from what we've talked about. Increase pregnancy rates and increase production and genetic gain by using genetic material from superior animals. So if you can ensure, basically what they're saying there is that the embryo is fit and healthy, you can use that one. You can detect whether or not it's, if it's not a healthy embryo, then you may choose to get rid of it. So that's an important biotech as well to ensure the maximum yield, I guess, particularly for beef cattle or dairy cows. Sperm testing to identify percentage of live cell counts and the percentage of degenerate sperm cells. So degenerate here means kind of faulty or unusable cells. You can use this data for sire selection and identification of sire fertility. So how fertile is the animal you're using to breed? If you can prove or find out that your bull, for example, is highly fertile, you're going to increase reproductive rates and increase production. Embryo splitting, spoke about earlier. Artificial embryo splitting increases genetic gain through splitting embryos from genetically superior animals, which leads to increases in production. So if I know I have the best genes, I have the best embryo, the cow that I'm going to produce from that is going to be superb. Well, it'd be amazing if I could split that embryo and produce two identical twins that have the same traits, and then I'm getting two cows with the best traits. So I want you to commit a few more of these to memory. BT cotton just seems so obvious. Obviously, a biofuel would be great as well. And the embryo, sperm, and embryo splitting they all make sense and maybe not too difficult to commit to memory as well. Okay, other biotech. So this is from the Master Ag Notes. So a little bit of repetition here, but it's just another way of saying what I've discussed there. So biofuels is the name given to fuels produced by the actions of organisms on organic material, usually plant material, by fermentation. E10 is a biofuel, 10% ethanol, a natural alcohol obtained by fermentation of sugarcane and or maize, and 90% octane. The benefit of biofuels is reduced reliance on fossil fuels, which is a finite resource, and ideally production of greenhouse gases. Ethanol is cleaner burning than octane. Biopesticides, plants that have been genetically modified to become allelopathic, that is, release chemicals that kill or hinder surrounding competing plants. This is called a biopesticide, as it does not involve the use of chemicals, but uses a natural plant survival mechanism, allelopathy. So that's a new term, allelopathy, but if you want to use that, that's awesome. But we already kind of know BT cotton, we've discussed this at length. It's just another way of saying it. Rumen Modification, this is a different example from the one to reduce methane. Involves the injection of bacteria by drench into the rumen of cattle, which work to detoxify toxins and acacias. This allows for cattle to feed on acacia or wattle, benefiting from the mass amount of protein in the feedstuff. So what we're doing here is this injection allows the cows to eat other foodstuffs other than what they're used to by detoxifying chemicals in acacias. So that's another cool example. Gene markers. Gene markers, fluorescent tags, can be attached to particular gene segments to allow for ease of identification in DNA analysis. This is useful for determining whether or not an organism has acquired a desired gene. Embryo and sperm testing. Biotechnological advances have allowed for the sexing of semen and embryos. Semen are sexed by placing the semen into a dye that stains X chromosomes bearing sperm different to Y chromosomes bearing sperm. So XX is female, XY is male, thus allowing for the sperm containing the respective sexes to be separated. Embryo sexing involves the extraction of genetic material and amplifying a segment of DNA on the Y chromosome, if one is present, by polymerase chain reaction, so PCR reaction. A test is then performed to determine whether the Y chromosome is present or absent. Currently, these methods are inefficient and very expensive for commercial use. And the last one they talk about in those master notes is the embryo splitting. Using micro tools under a microscope, embryos are cut in half to produce identical twins. That is, two embryos with identical genetics. This effectively increases the number of offspring produced from a valuable donor with desirable genetics. The embryos are then implanted into recipient animals. Awesome. A lot of information there, guys. Some maybe more examples that we haven't discussed, which is cool. Put them in there. The Rumen Modification one. The obvious one is reducing methane, but they've got one there that allows them to eat more diverse foodstuffs as well, which is cool.